Should we start or wait?
मेडिकल में स्टार्ट करना चाहिए
2:09 हो गया.
टाइम का कोई है जो टाइम का ध्यान रखेगा. अच्छा तो I think it's 8 but we can start.
Yeah, I think so.
Okay, good afternoon.
Sir, I'll.
Okay.
Well, it is. Well, I welcome you all to the session on emerging infectious diseases. It is my honor to introduce the chairpersons for the session, Professor R.K. Seth. Professor Seth has been the professor at the Department of Zoology for the longest time. I had the pleasure of attending his farewell just a couple of months ago. So Professor Seth's research interest lies in applied entomology and radiation biology. Professor Seth did his PhD from Department of Zoology, University of Delhi. He has been an advisor on a lot of important committees of Government of India, ranging from Ministry of Environment and Forest to some staff selection commission. He was also awarded a postdoctoral fellowship by European Commission, which he pursued at the University of Bath in the UK. Welcome, sir. Our next chairperson for the session is Professor Sanjeev Kumar, who is currently at the Department of Biotechnology, Chaudhry Bansilal University. His research interests are in molecular and vector biology of malaria parasite. Professor Kumar did his PhD from Banaras Hindu University, and he's a recipient of several awards from NIH USA. So now I request both the chairpersons to initiate this session. Thank you.
Should we start? Okay, good afternoon. We are going to start this session, and I would request to stick to the time to the extent possible because we are slightly late. And our first speaker is this is the keynote lecture by Dr. Anant Narayan Bhatt. I'm very pleased to introduce this gentleman, scientist F, and he's in INMAS. I have got a very wonderful association with INMAS. For the last 30-35 years, I have been using Gamma Cell from INMAS. What's up? Okay. So, his area of interest is biochemistry, cell biology, radiation biology. Radiation is one common factor over there. People have been using nuclear energy for various purposes, for medicine, for agriculture, for diagnostic purposes, etc., etc. And emerging medicine, development of prophylactic, mitigative, therapeutic drugs for nuclear and biological emergencies. And he has a wonderful academic record, research record. He has been visiting scientists in Switzerland. He has been visiting Fulbright Scholar in USA. So with all this brief introduction, I would request him to present his talk.
Thank you very much to the chairpersons, Professor Seth, who have been our guide and mentor in radiation biology. As he said, we had long collaboration with Delhi University Zoology Department, and I have privilege of teaching also as a guest faculty in Zoology Department. So, and I'm thankful to the organizer also who gave me this opportunity to present my work here. And the work is 2DG as anti-COVID therapeutic from an age-old molecule to a modern broad-spectrum antiviral therapy. So this is the work which we have done during the COVID. Being in a defense institute, we have been always, as radiation biology, as I said, and this molecule has come basically from there only as a adjuvant to adjuvant to cancer radiotherapy. We repurpose this molecule as a anti-COVID drug. So, I mean, before going to that, I would just like to tell a little bit about the basic, I mean, how the biochemical pathways plays a role in cells, and then how they can be a very good target. I mean, you must be hearing a lot about that. What are the biochemical pathways and what can be, when they can be targeted and when they cannot be the target. When in differential systems, and like in cancer, they are differentially expressed. In diseases also, they are differentially expressed and they can be the target. So I just want to connect this. This is from physics. You all have heard in basic physics the Kirchhoff's law. I mean, the current coming I1 plus I2 is equal to I3 plus I4. But long back, one of my friend shared this image, maybe somewhere in, you can assume like somewhere in Delhi, in the city. He asked the question, can you apply Kirchhoff's law to this circuit? So it is very difficult circuit. It is very difficult to find out current is coming from how many sources, from where, and then current is being distributed to how many different circuits. So this was a very relevant question, but you must have known that this circuit is even more complex than that circuit. Right, whatever we have seen, a difficult circuit to find out that how much current is going in which direction. This circuit is even more complex, which is going on in cell every fraction of seconds in our every cell in the body. And the tribute goes to all those biochemists, all those scientists who really solved this circuit, and they know like when a molecule enters in the cell, where does it go, how does it benefit to life, how does it manifest in the form of life, and that's how we can, think that probably some of them can be the target and some of them can be utilized for that. So one of the old scientists, Otto Warburg, you all know right now the biochemical pathway for energy. Cancer cells basically rely more on glycolysis when they have to take the energy. This was an understanding long back in 1930s when he did this work, and he found that cancer cells have compromised mitochondria. Then he saw that what basically when the mitochondria is compromised and energy is basically, mitochondria is known as powerhouse of the cell. So these cells are taking energy from where? Then he realized that these cells have high glycolysis. And this phenomena is known as Warburg effect. In cancer biology and in biochemistry also, it is known as Warburg effect. Why Warburg effect? Like partial effect in the absence of oxygen when glycolysis goes up. But even in the presence of oxygen when glycolysis goes up, it's called Warburg effect. And this is a very well established phenomena. And what basically it does, so this was the background of that anti-COVID drug which I am talking about. That normal cells, when some cell gets damaged, to survive, they change their metabolic pathways, and that change in the metabolic pathway give them impetus. They start proliferating very fast, and that's what happens in cancer cells. And when they are proliferating very fast, the glucose demand goes very high, and that's what happens. And 2-DG is one of the drug which is anti-glucose. It is exactly like glucose, but at the position 2, the hydroxyl group is not there. So when you go in the glycolytic pathway, it gets converted into glucose 6-phosphate, 2-DG 6-phosphate, but that cannot be converted into fructose 6-phosphate. Why? Because at position 2, oxygen is required to convert a glucose into fructose, right? So because oxygen is not there, it cannot be converted into fructose 6-phosphate, and that's where it gets trapped inside the cell. It doesn't provide energy, and that leads to several other problems. So why glycolysis? Because single carbon metabolism, when the metabolism is very fast, what happens, the glucose input goes to glycolysis as well as triple P pathway, right? So triple P pathway is mainly anabolic pathway which gives you nucleotide metabolism, which gives you single carbon metabolism of fatty acid, as well as protein also it gives input. So when these things are required at very rapid pace, then the cell rely more on glycolysis. I will give you example. When we are in adrenaline gets secreted, when we are in a situation where we are in an emergency like fight, flight and 3F hormone, we rely mostly on glycolysis. Why? Because glycolysis can give energy at very fast pace. It is not economic, but by using lot of molecule of glucose, you can produce energy at very fast pace, as well as this anabolic reactions also at very fast pace to come out of that emergency situation. So that is why glycolysis is important. This is the background. I mean, long back since 2 decades, we have been working on this. And these two gentlemen who were our seniors, former director, Professor Vinay Jain and Dr. B.S. Dwarkanath, he also had very long association with Delhi University. They worked on this molecule as a adjuvant to anti-cancer therapy, giving just before radiation, so that when radiation kill the cells, damage the cells, the energy will not be available to repair the cells. So the damage will be potentiated and the cancer cells will die more. This was the hypothesis, and it can arrest also the cancer cells growth. It will not kill the cancer cells, but it will arrest the growth of cancer cells. So the tumor will not go further. This was the hypothesis, and they used it, and the clinical trial was also conducted at so many hospitals in glioma, glioblastoma patients. So it was already used in humans, and later in 2014, DCGI has given approval, recommended this drug for anti-cancer as an anti-cancer molecule. These are some of the clinical trial reports which they worked on and then published in a very high impact journals, the clinical trial reports. And this is some of the recent work where we use a low dose of 2-DG to establish that if you give 2-DG initially while cancer is progressing, then cancer can be arrested at that initial situation, and this can be a very good drug for when carcinogenesis is induced. As I said in the beginning, that at the time of initiation of the carcinogenesis, metabolic switch happens. So if you target at that point of time only, probably we can stop the growth of the tumor there itself. But today we are going to talk about the antiviral, how it works as antiviral molecule. So, like this was already established in the literature. It started coming sometime back only. One clinical trial happened in 1983 in herpes virus, where 2-DG was used topically. I mean, because herpes happens on the skin. So it was used topically. Somehow it didn't work. That clinical trial was done in 36 patients, but it didn't work. So that work was stopped at that point. But recently, a lot of basic research was going on, and people have shown that when virus infects the host cell, because virus also needs very fast production of the virion molecules, right? A lot of virions. So that is also proliferation at very rapid speed, and that also needs similar glycolysis kind of switching from mitochondrial respiration. So that is why virus on infection, it hijacks the host cell metabolism. And there are a lot of reports, and recently in 2018, this paper got published in PNAS that rhinovirus, when it infects to the host cell, it basically switches the metabolism of the host cell, and probably that host cell metabolism can be targeted to as an antiviral therapy. So this is the problem in SARS-CoV-2. We all know that this is a very highly infectious virus, not like any of the other viruses. This was very highly infectious. And when Omicron came, that was 18 times more infectious than the first virus. So when we are talking, if I am infected, it will go and then you will respire it, you will inhale it, and then this will create the problem. So this is the entire etiology. I think most of us, this is one disease probably even a layman also got a lot of experience and knowledge about the disease, right? So these are the 3 different stages. Stage 1, when it reaches to the lung or even oropharyngeal region, and then it multiplies very fast there, and the virus multiplication happens at very fast rate. Stage 2, when it reaches to the lung cell, and you can see in the bottom how much damage to the cells it is causing. On the left side, it is uninfected cells, and on the right side, you can see the infected cells. So cells start dying. They will start, that is called classical in virology, it is called cytopathic effect. So cells will start dying because of the multiplications of the virus is happening at very rapid rate. You can see here. And this is what damage. And when the cells will die, what will happen? Inflammation will take place. So cytokine storm will coming into this stage. And in the third, you can see here the stage 3, the damage has taken place and lung has completely gone. I mean, because of inflammation, a lot of fluid comes in and then accumulates, and that is the situation when recovery of the patients become very difficult. People are shifting on the ventilator and they are dying. So these are the 3 different stages. How 2DG can work in which direction? You can see here that the virus multiplication is coming out from the cells is very fast, all these viral molecules. And what basically this has? It has only 3 molecules, I mean lipid, protein and carbohydrate little bit, because the modification of protein is happening, and then nucleic acid. So these proteins are phosphoproteins, so they need ATP, glycolysis is required there. Nucleotide synthesis, I told, like that is also need glucose there. And then even lipids also need a single carbon synthesis. So that is why for fast viral replication, it needs a lot of glucose here in this molecule. So what basically happens inside the cell, you can see here, this is a normal cell. It is most of the glucose when taken in, it goes to the TCA cycle and then normal process goes on. However, in infected cells, what happens? That once it infects, it damages the mitochondria. Viral infection damages the mitochondria. It actually basically nitrosylates the mitochondrial protein. So majority of the mitochondrial protein, they will not be able to perform normal respiratory functions. So not completely damaged, but partially damaged the mitochondria. And that allows more viral infection. Actually, it is a vicious cycle. So what happens that during that situation, if cell has to survive, or the virus reprograms the host cell metabolism by integrating its genes, and that's what it does. Lot of glucose transporters will increase on the surface. So it will invite more and more glucose to get accumulated inside the cell, which will be utilized for more and more viral replication. So this is basically what happens in case of viral infection. So on stage 1, you can, by targeting 2DG, you can arrest there the viral replication. In stage 2 also, because inflammation, and from anti-inflammatory to pro-inflammatory, the inflammatory cells also become more glycolytic. See, if you inhibit the glycolysis, macrophage can't do phagocytosis. So glycolysis is intricately required for even inflammatory functions also. So if you inhibit glycolysis in inflammatory stage, it will inhibit the inflammation as well. This is a very well established phenomena in the literature. So you can go and check that. So, so this at stage 1 and stage 2, both you can arrest COVID. So this is the molecule you can see here. I mean, this is simple glucose molecule. At position 2, both are H and H. So this becomes very close to mannose also. I mean, mannose and glucose, they are epimer, right? At position 2, OH is below, and in mannose, OH is up and H is down. So this is how this acts as this mimics even mannose also. We will tell why mannose is important in later part of the thing. So it is exactly at C2 position, only the oxygen is gone. So it behaves exactly like glucose outside the cell. I mean, body cannot differentiate between glucose and 2DG outside the cell. But when it goes inside the cell, as I said, it will get arrested there, and that's how the problem starts. I mean, it will arrest more in cancer cells or virally infected cells. We will show here. So as we discussed already in detail, that upon viral infection, it hijacks the host cell metabolism, and then the cells become more greedy for glucose. A lot of glucose is required. And like in infection also this happens. Infection leads to insulin resistance, and more glucose comes in the bloodstream. And that's why more glucose comes in the bloodstream, because it has to facilitate the macrophages and inflammation to sort out, to come over that infection. And this is a natural phenomenon, happens in all of our body. So this is the thing, like glucose more glucose requirement is there at that point of time. Then as I said, like ATP, rate of production of ATP is much faster in glycolysis. Like if a single molecule of glucose, let's say, take 1 minute from going from top to bottom, I mean conversion to carbon dioxide and water through oxidative respiration, it will produce, let's say, 36 molecule of ATP, whatever as per the classical biochemistry. But if in the same 1 minute, even if it is not going to the glycolysis, to the mitochondrial respiration, if glycolysis is high, 50 molecule of glucose can be processed. So what will be the total net gain of ATP? That will be 100 molecule, right? So as compared to 36, you are able to get 100 molecule of ATP. So that is why in emergency situations, glycolysis is basically required. When we are running, glycolysis is basically required because oxygen is also not available at that point of time. Then other than that, I mean, SARS-CoV-2 infectivity, metabolic plasticity is very important in normal cells. It can switch over between the glycolysis and mitochondrial respiration. But in case of virally infected cells, as I said, virally infected cells and cancer cells, mitochondria is damaged. So glycolysis is the only pathway which is providing energy in that to do any function, either to synthesize the virus or to survive the cell. So targeting becomes easy. And these are the very initial evidences. We also started our work in March 2020 when COVID came in India. And, you know, this paper got published in May 2020, suggesting that they have done simple proteomics of, they infected the host cell, and they say that what are the pathways which are getting activated, and all these pathways can be targeted. So this is a very basic work in which they found glycolysis is also upregulating, so glycolysis can also be targeted, and they showed also 2DG, using 2DG, the viral replication is going down. So here, and this is another work which will suggest, I mean, in common public, we know diabetes were more susceptible. Why diabetics were more susceptible? Because they have high level of glucose continuously circulating in their body. And this paper published in Cell came little later in 2020 end, which suggested that if glucose level will be high, it will facilitate the viral infection in the cells, and that will lead to more and more, I mean, SARS-CoV-2, the body will facilitate the SARS-CoV-2 more infection. So these two work got published, and parallelly we were also doing, this we already discussed about. And then coming to then what we have done, how we found out that probably 2DG can be a good molecule. So these are the results. You can see here that this is uninfected cells and this is infected cells. This green color is a fluorescent 2DG, you can say, because it's a fluorescent molecule. And you can see here in uninfected cells, the accumulation of 2DG is not very high, but in infected cells, accumulation of 2DG is very high. This is the quantitative data, and you can see here the glucose uptake and lactate production and ATP production also goes very high when the infection happens, right? So 2DG becomes, and upon infection, you can see all the GLUT1 goes very high, which can trap the glucose at very low level of glucose. These are very efficient glucose transporters. At low level of glucose, they can transport, and GLUT3 as well. So the level of GLUT1 and GLUT3 goes high, GLUT4 also goes high. So they all are pushing more and more glucose in the virally infected cells. And then the enzymes also, which are processing glucose, that has also gone up. So you can see here that when we infected SARS-CoV-2 in Vero E6 cells, we found that all the glycolytic markers are going high. And this is a very important thing. So people say like this is a normal glucose, it will go to every cell. But you can see here this experiment of us, we co-culture two cells. The one cell which takes very low, very normal viral infection, that is Vero cells, they are isogenic cells, and the Vero E6 which is modified and it takes high viral infection. So these Vero cells are labeled as red, you can see here, and Vero E6 cells are not labeled. So we have given, they are co-cultured, and then we have given them 2DG. You can see that all those cells which are not looking red, they have accumulated more and more 2DG, right? So this suggests that even in lung cells, when some cells will be infected and some cells will not be infected, it will be mainly infected cells which will accumulate very high amount of dose. And in drug, when we are designing drug, this is a very important thing. The drug is targeted. I mean, this is using the inherent phenomena of the body or the body mechanism to target the drug. We are not doing any other targeting. We are not designing the molecule in such a way that it should go to only the targeted cells. It is using the inherent phenomena to reach to the target cells only, and that is what basically happened here in co-culture. So in a simple model we have tried to explain here. You can see here that these are only virally infected cells and in lung cells, and it is getting accumulated more as compared to uninfected, where the level of 2DG is very less. And you can see here that as compared to uninfected, this is the quantitative data that the 2DG is very high. Then the other things come, because you know in cancer biology people have been thinking that 2DG is to kill the cancer cells. So they said that probably 2DG will kill the other cells also, which is not required probably in biology. But it is very well known that it will arrest the growth of the cells, and our normal cells are not proliferating, right? Our blood cells are only proliferating. Some of the cells are only continuously proliferating. The rest of the cells are senescent, I mean they are not proliferating. So it inhibits the growth of those cells, which we have shown through here, that it is not basically killing. Clonogenicity is also not changed, and it is basically not killing the cells. What it is doing, it is just stopping the proliferation of those cells. Then we checked its inhibition. This work was done during lockdown. It was done in May when I went to CCMB and conducted these experiments, because that time virus was only available in CCMB and NIV. These were the two institutes. So we conducted this experiment at CCMB in B.6 variant, the very first variant which was detected in India. And we found that the effect is very good. And later we did in B.1.1 variant, which we established in our BSL-3 lab. First we established the BSL-3 lab at INMAS, and then we established these cultures also. We found that this is equally effective here also. In Delta and Omicron variant also, it is equally effective. And you can see here the production of the proteins also. I mean, the N protein, nucleocapsid protein and spike protein also goes down when the 2DG is given to the cells, infected cells. Then in fact, you can see here, very well, you can see here this is an infected lot of cell death you can see, which is called CP. Classically in virology, it's called CP. And at 1 millimolar and 5 millimolar, you can see here the effect is completely gone. How much time I have? Okay, I'll try to complete quickly. So what I said, you can see here with increasing glucose, like in case of diabetes, people thought that probably 2DG will interfere with glucose and effect will go down. But you can see here that at a very high concentration of glucose also the effect is not compromised. 2DG effect is not compromised, but mannose is able to compromise its effect. Why? Because mannose at very low concentration it is taken up. So 2DG at very low concentration it will be taken up and it will be able to affect. So one hypothesis was directly by inhibiting glycolysis, by anabolism, it is able to sequester the virus. But you can see here what we found in the next, when we have taken out the virions from 2DG treated cells and untreated cells, and we calibrated, we equalized the concentration and then infected without 2DG, you can see the virions are not able to infect the new cells. Why this is happening? What is the reason? You can see here that 2DG it mimics mannose, and mannose is required for what? Basically for glycosylation of the protein, and all the proteins on the virus surface, they are glycosylated. So and glycosylation is like address mediator. So if you write a wrong address, it will either not reach to the right place, the letter will not reach to the right place, or it will be again go back to the post office. So this is what basically happens. You can see here the two glycosylated moieties become deglycosylated, and that is why probably virus is not able to bind. So this is, we proposed a model, and you can see here the virions are not able to, the virions produced from 2DG infected cells, whatever, like let's say 90-95% inhibition is there. So whatever 5% virions are being produced after initial 2DG treatment, they are not able to infect the neighboring cells. And that is what we proposed in this. This is a common normal thing which is happening. But in 2DG treated cells, first thing it will reduce the viral production, and whatever virus will be produced, they will be with deglycosylated protein, so they will not be able to infect the neighboring cells. So overall, it is able to infect. Stop none of the other drugs. They have two mechanisms of working. They don't have even direct targeting of the cells, but this drug does have. This study got published in Life Sciences. And as I said, like this has a very good anti-inflammatory effect also. So it will be able to capture at two stages, stage 2 of the COVID also. And this is the PNAS study which got published, where they showed that 2DG works very well in rhinovirus also. So this we already explained. Based on this, we did clinical trial. We went to phase 2 and we did clinical trial. And all the patients who were recruited in this clinical trial, they were actually at stage 5. WHO has given an ordinal scale. So 5 means all the patients who need oxygen. They are not only hospitalized to the hospital, but they need oxygen supplementation also. That means oxygen is below 94% saturation. So they need oxygen. So all such patients were recruited. And when we given them different doses of the 2DG, we found that 90 mg per kg per day is very effective dose. And you can see here, this is the parameter. This, like time to achieve SpO2 94% was from 5 days, it has came down to 2.5 days. And then time to discharge also reduced, time to clinical recovery also reduced, and then time to vital signs normalization, time to achieve 1 point on WHO ordinal scale, and time to achieve at 2 point at WHO ordinal scale. Both have gone significantly down. Based on this, actually, we have gone to phase 3 also, and we performed this study in 220 patients. These are some of the key things, and you can see by day 3, significant number of people is coming out of showing 2-point improvement, and this is 1 point improvement, which suggests the requirement of oxygen, the damage to the lung cells, which promotes the oxygen requirement at that point of time is basically that is working very well. Anti-inflammation is working, it is targeting inflammation very well, and the drug is able to give relief to the patients very nicely. And this is the ICU patients you can see as compared to these red line. I mean, we compared everything. This was used as an adjunct. So in control, all the patients were given remdesivir, favipiravir, all the drugs. In 2DG treatment, they have given along with these antivirals, 2DG has been given, and you can see as compared to them, the patients coming out of ICU is very fast as compared to a standard of care where all the antivirals have been given. So this study also got published recently in BMC Infectious Diseases. And, 21, this got the emergency use approval from DCGI, this drug. And it is available in the market now, and it is very simple. I mean, it comes as a glucose powder, so you can sachet you can dissolve in 100 ml of water, and the people can drink it. And these are the people who contributed immensely. Like when we do research in the lab, it is just like we only have to manage budget and everything. But when we were going to industry, we had to open one pharmaceutical industry, Dr. Reddy's lab. So Dr. Reddy, actually, our chairman and secretary DRDO that time, he is the person who converted this possibility into reality. He is the man behind this who helped us a lot, and meeting all the challenges, funding and everything. Thanks to him, and then all the scientific team and my students, during the lockdown, labs were not open, but we had got requests and then permission. So all this work was going on during the full lockdown also in my lab. So thanks to all the students and my collaborators who worked well. And this is the team from Dr. Reddy's lab who also worked day and night during that time to bring this molecule to the public. And finally, this got approval in May 21 to use in emergency situation in hospitalized patients. And I'm happy to share, actually, I'm carrying the pouch also. It is very privileged for any scientist, like your research from the lab, it comes to public to use. So this is. Thank you very much.
They have some questions.
I think we are running short of time.
One or two questions I think we should have.
Yes.
What is the effect of 2G and RBC?
RBCs. See, we have not checked, but as you said, like rightly, we have been thinking. I think you are asking from glycosylation, deglycosylation. Yeah. So
They directly depend on
glycolysis, right? So
they carry oxygen.
right. So as far as this oxygen carrying is concerned, that was one of our main parameter. So that got even better. But our main concern is what happens if glycolysis, it is able to modify the glycosylation. I mean, RBC only carries A, B, O blood groups, right? So our main concern is from side effect perspective, from that perspective, and really we have not checked. As far as the number of RBCs and number of WBCs are concerned, they are absolutely normal in the control patients as well as treated patients. So this is the clinical parameter I'm talking about. We have now more than 400, 500 patients treated with 2DG and they are absolutely fine. I mean, there is no problem in as far as the numbers are concerned. But yes, I mean, that concern is already there. We are working on in that direction to check whether there is some glycosylation changes are happening or not because of continuous use of 2DG for a long time, like 15 days or something.
And also, like, caloric restriction is good idea.
Right. For cancer, it is very nicely working. Right. Right. So what work I have shown you published in European Journal of Cancer and PLOS One, there we are using it as a dietary supplement. 0.2% continuously in water. The mice, they drink that water continuously whole day and tumor formation is much less. I mean, first lot of mice, they don't develop tumor on implantation. Those who develop, they develop very small tumors. So that is a very, I mean, that work is already published in PLOS One and European Journal of Cancer. You can check, search on the Google and you can find it, that study. So it is working very nicely as a dietary supplement.
I think there was another guy. You wanted to ask something?
Yeah, please.
Can this 2DG differentiate between the infected cells and non-infected cells?
Yeah, that's what I showed one of our experiment where, as I said, like the transporters on the surface will be very high. So in normal cells, only very few transporters will be there. So it will go there also. But as compared to uninfected cells, when infected cells, they have very high number of transporters on the surface. So they will accumulate much high. The other thing is, like when we give 2DG, whatever dose we are giving, it reaches only 0.5 millimolar to the blood, right? But glucose is already 5.5 millimolar in the blood. At 100 mg per deciliter, it is 5.5 millimolar in the blood. So it will not, in normal cells, it will not have any problem. But in infected cells, the transporters which are expressed on the surface, they have very high affinity with the glucose. So they will sequester more and more 2DG. So it will go mainly to uninfected cells. Infected cells. Right. Yeah, that's what I showed. Yeah, I showed a data. I showed a data. I mean, we have gone up to
almost like Equimada, right?
Right.
One is 500.
Right. No, it is 0.5. 2DG at the dose which we are using, it reaches 0.5, almost 9 to 10 fold difference, 10 fold lesser than this. So when we showed at 10 millimolar glucose, then also it is able to inhibit that viral replication. So we in media we used 10 millimolar glucose and we used the same 15 millimolar glucose and used the same 5 millimolar glucose. In vitro I'm talking about, it is able to inhibit the viral replication there also.
Dr. Anant, Dr. Anant, on this side.
Yes, sir.
So 2DG is a natural molecule or is it a man-made molecule?
Sir, it is synthetic man.
Synthetic.
Right.
So not found in nature.
Sir, it is found in nature, but that position 2 is methylated. It is believed that some of the fruits they do have this molecule, but at position 2 there is methylation. So there also it works like the same as it is working here, but this is a synthetic molecule.
Can you name those fruits so people
Actually, I'm also searching for that, but I have heard that there are some herbs which people in Brazil and South America, they use it as a tea. So I'm also searching. I just have heard that they have similar kind of dietary effects, but I don't know exactly.
Thank you.
Right. Yeah, anything which gets methylated at position 2 may work like that.
So in fact, Dr. Anant, you are the real hero behind the real situation, because you have a number in a COVID situation, and 2DG all of a sudden came in picture. And thanks to God and thanks to you people.
Thank you. Thank you.
I first heard this name to DG from Surya.
Yes, sir. He is the father of this.
Now, I would request Professor R.K. Seth to kindly felicitate Dr. Anand for delivering such an exciting lecture.
Can I also have this pleasure to come in?
Of course.
It is such a real hero. I think.
Of course.
I need a picture.
So now we are moving ahead, and our next speaker is Professor Lalita Gupta. She is from Chaudhary Bin Salman University, Bhiwani, Haryana. She had done her PhD from Banaras Hill University, and afterwards she has postdoc fellowship at COML France, followed by Colorado State University in US, and then National Institute of Health, NIH, USA. And later on, she had been a professor at BITS Pilani, and then after that, she's at CBLU. So she's working on the mosquito immune system and interaction with the Plasmodium as well as dengue virus. So I would like to invite Professor Lalita to the platform. Professor Lalita, please.
Professor Seth, Professor Sanjay Kumar. It is my great pleasure to be present over here. I would like to thank the organizer, Dr. Alok Hardy, for giving me this opportunity to present my research. Here I'm switching from COVID-19, which is an excellent work done by Dr. Bhatt, who was here, to mosquito immunity. So it's like a kind of switch. And I hope you were able to understand that though we recently gone through this COVID infection, mosquitoes and the diseases transmitted by mosquitoes are also very, very important. During that time, I remember, like when COVID is going on, so much, like so many dengue infection is going on. It is very difficult to find it out. It's really a dengue infection or it's like a COVID infection. Everybody was like kind of scared. So today, what I'm going to present over here is about the transcriptome analysis of ATC transporters in immunity against blood-borne pathogens. So before I would like to give some introduction and why we are working over here, what is ATC transporter and what we know about mosquito and its immunity. So just now we have learned about like we human are vulnerable of different diseases. Here we talk about direct infection, which is like a COVID. We just learn about how this COVID is affecting us. Here are some blood-borne pathogens where we talk about indirect. Here we require two hosts. So when we talk about two hosts, like here, in case of blood-borne pathogens, we are talking about the one host is mosquito and the other host is the human. So I'm giving you two pictures just as a glimpse. Like here it's a malaria, which is transmitted by Anopheles stephensi, and there are several species of Anopheles. And on the other side is a dengue, which is transmitted by Aedes and many species of Aedes. So there is always easy to target the human host. But when we talk about the mosquito host, it is important because if you think about, like here, I'm giving just examples of several other species which transmit the disease and even killing the people. Mosquitoes is one of them. And because they're able to transmit dengue, malaria, West Nile, chikungunya, and variety of bacterial and viral infection and plasmodium infections. So I would like to go why we are why we are looking for mosquitoes. Because when they transmit several diseases, there are different ways to control it. One way is control, it's just for protection, as we all know that we can get vaccinations. On the other ways, like we have our different anti-malaria drugs and other drugs which we can protect us. On the same time, we just looking at the transmission blocking. So if we able to block the transmission from mosquito host to the human host, that will be easy. So we can not allow parasite to go inside the mosquito. In that case, this term we use as a transmission blocking. One way we can just even kill the mosquito through the insecticides. On the other way, we can prevent ourselves, just we are using different, like bed nets we are using Revlimid. And one of the very important and emerging one is like a blocking parasite development inside the vector. And when we say inside the vector, we have to know more about vectors. So I'm just giving you first two glimpses of one. I worked on like a little bit on killing the mosquito because this is easy, we're able to do experiment very easily. And then we move towards the blocking the parasite development. So you can think about the mosquito immunity and how they able to control parasite development inside the mosquito. So just giving a one insecticide example. In case of that, we were looking for when we came from US and Wits planning, like we were just like setting off mosquito lab is not very easy. So we were setting the mosquito labs, same time the parasite and everything, and at the same time we were saying, okay, let's look for the any biological way which are environmental friendly, we can control the mosquito population. So here I am giving the one example of Agave agassifolia. This plant is present in the Rajasthan area and towards the desert, and very specific that when we take just water extract, they have a efficacy to kill the mosquito larva up to 100% within 48 hours. So, and important thing is that they give the activity only in time of summer. As soon as winter comes, no activity. So we were surprised that the student might not be doing correct experiments. All of a sudden it's required that we thought, okay, let's change the temperature. So when we change the temperature 37 degrees, again this plant extract is being released. So we published over there in the journal Frontiers, and now even government is also wants to know about this plant, and they are looking more how we can find a different extract and use it for just killing the mosquito. From here, I'm just switching towards the mosquito immunity. So once we know how the mosquito immunity is able to kill the parasite, we can easily block the transmission. So mosquito have a very strong immunity. One is called pattern recognition receptor, so its innate immunity is present, and they also have a signaling molecules like immune signaling pathway. I'm working on JAK-STAT pathway. So JAK-STAT pathway is very well characterized now in mosquito, and we found that this JAK-STAT pathway is actually working as a negative, positive regulator. It's actually suppress the plasmodium development. So just for students who are not in this field, this plasmodium, they enter into the mosquito, and it's almost takes 21 days to stay inside the mosquito, and the mosquito is able to transmit the disease. And we are looking at the stage because this is the epithelial, and we are looking in the stage when it's become the oocyst. So it's better to, if it is possible, we can control this transmission because it is a midgut epithelial, And if we control over here, that is much different. So I'm just giving an example of STAT signaling pathway. We silence the STAT molecules, and when we silence the STAT molecule, we find it out in case of energy tertiary. We find that this is increasing the parasite load. That means we calculate on each individual mosquitoes the number of poses present over. On the other side, we find it out that in day 7, when we talk about the day 7, it's like of SOX gene, which is the suppressor of STAT pathway. This is going down. That means it's suggested that STAT pathway is activated, and when we silence, that the parasite number is going up, and when we have a SOX, then the number is going down. And this interesting finding, we found out that not only the early stage of poses, but even the late stage of poses, this STAT signaling pathway. So from here, we are started looking more about the STATs, and here we find it out like there are several STAT molecules are present in 18 Anopheles mosquitoes. Some having a STAT A as a conserved one, which have a different introns, whereas a retrotransposon there is a STAT B. And actually STAT B is regulating the STAT A. But in few mosquito species like Anopheles sinensis, Dirus, Dilinity, Albinus, and Ferropi, they have a single STAT. So when we talk about any like control mechanism, and we say it's easy now to find it out molecules and can be easily controlled, we find out there are so many types of Plasmodium present, and there are different in mosquitoes. So combination of all these to have a mosquito and parasite and finding out one target molecule still is going on, and still we are looking for. After that, we move to one of the molecules which is present inside the mosquito, and that is also an immune molecule, that is apolipoforin. And here we also do the silencing experiment. It's a big experiment. It takes like almost 12 days because we have to use the double-stranded RNA, inject inside the mosquito, give the Plasmodium infection, and allow the mosquito to survive, and then started counting the parasite. So I'm giving here the example of apolipoforin 3. This is known for flight lipid proteins, but they are also play a very important role in mosquitoes. So here when we see that black C and when there is a apolipoforin 3, the infection is going down. So that is actually working as a negative regulator. So when there is a no apolipoforin, parasite number is going to be increased. So it's actually helping parasite to grow. So here I'm just saying that we are working hard to find out few molecules which going to target towards the Plasmodium development inside the mosquito bag. And in that case, we are finding new and new molecules. Now I'm coming from here towards the ABC transporter, which we recently started working. And these ABC transporter, why we started working? Because we find without some ABC transporters, they are actually having a role in insect immunity. Some ABC transporter immunity, these are the sample bursts which coming out. And from here, we also search the literature, and this we find out that some, like in case of wheat or in case of other molecules, they are actually transporters are sensitive to or suppress the pathogen development. And single mutation in Bombax gene, we find out that with that ABC transporter resistance to the BT toxin. And there are a few more examples where we say yes, these ABC transporters are working. I'm just giving enough time. Yes. Okay. So here we would like to know how this ABC transporter, which says ATP binding cassette transporter, and whether it's working in mosquito immunity. So this project was funded by DPT, and we're looking for some novel immune molecules which may regulate the pathogen development and might offer a new insight to interpret the mosquito cycle of bloodborne pathogens. I'm giving this example how this ABC transporter looks like, and in Aedes aegypti. So ABC transporter have the two domain, one is called nucleotide binding domain and other one is transmembrane domains. This nucleotide binding domain is actually having a ATP binding, like you can see over here, and they have a transmembrane domains. So transmembrane protein that transport amino acid, sugar, peptide, small movement across the biological membrane. And as I said, they have a NBD, which having a Walker A signature sequence, Walker B motifs, and they have a signature sequence. And TMT, they have a 6 helical embedded in the bilayer membrane. So this is a ABC transporter proteins. Based on how many ATP binding acids they have 2. One is called half transporters, or we can say full transporters. You can see this is the half transporter and the full transporter. This is the mechanism how it working. That molecule is coming, ATPs, and able to release the molecule over there, and it generates ATP. So what we are looking over here, so I'm talking about Aedes aegypti mosquito. And in Aedes aegypti mosquito, we are first identifying and characterizing the ABC transporters, and then we are looking for their expression in the mosquito development and identify their expressions and find with the immune challenge. We are dealing with the challenge of bacteria over here and finding out if there is any molecules. So reverse genetic approach we are using to find out the specific molecule of ABC transporter. So with the literature search and the database present over here, we are looking for ABC transporter, and in case of we look over here in Aedes aegypti, there are almost 59 ABC transporters are present, and they are having a subfamily from A to H. We compare like in Drosophila, Anopheles, Gamma, Bombax, Pulex, as well as Homo sapiens. What we find out that ABC A, C, and G subfamily are widely expanded. So we published this paper last year in Pathogen, and afterwards we are also started looking like whether this ABC transporters expressing inside the mosquito gut or in the carcass. So here are some genes which we say like in case of ABC. Some are expressing both in midgut as well as in carcass, whereas some are only expressed in carcass or some are only expressed in the midgut. So there are some very unique because they have a different domain structures over here. So we started targeting few of them. How we targeted? We are looking for some molecules, like here I'm giving the example of ABC B2, C10, D1, and G3. We did the experiment giving the live bacteria challenge with these mosquitoes as well as heat-killed bacteria challenge. First, over here you can see the different time kinetics from 1 hour to 12 hours, and looking the defensive, which is very known, molecule antibacterial molecules, and this expression is going high due to the relative mRNA expression. B2 of ABC transporter B2, this expression is there but not very high. Same way in case of C10 and D1 and G3. So this expression with this one, we can say that expression goes up and down in some cases, and here in some cases you can say in C10 or G3 the expression is like going up and then again going up in 6 hours. So from there we are looking for different. This is a little bit very big slide because we are looking at different ABC transporters and how these different families are working. So I'm targeting only 1, 2, 3 over here, and this is the 4th one and 5th and 6th. When we challenge, we say ABC transporter of subfamily A, C, and G are induced after both the bacterial challenge, if it is heat kill or if it's a live. Then we looking for one important one that is called ABC G3 because they have a similar E. coli toxin antitoxin system, and we find it out that how they are similar to other and where it is fine. So we use KEGG's sequence similarity database to analyze this ABC transporter G3. Now from here we plan to silence this ABC G3 genes through RNAi, analyze its effect on blood-borne pathogens. So here you can see that there's PCR amplification of ABC's G3 genes of 4 sets of midguts we are using over here, and then we clone it and make a double-stranded RNA of them. And finally, you can see the double-stranded RNA of AB6 And after that, we did the silencing. So here is the silencing results. We made the double-stranded RNA of G3. This is the control. There is a 80 to 90% reduction in mRNA level, and then we are looking for their bacterial load. So we find it out that when the gene is silenced, the bacterial load is increasing. In case of sugar fat, as well as when we give the bacteria supplements, also it is increasing. We suggested that ABC G3 is actually working against this mosquito, Aedes aegypti, and is working as an immune molecule. We look for another gene, that is ABC-C12, and we did this experiment to find out their viral load when there is a dengue infection. And from here, this experiment is preliminary experiment. We are also again looking to more details. So based on all these experiments, we are just giving that mosquito having a strong immunity, they're able to kill the parasite inside, but we have to find it out exact molecules for the different targeted molecules over there. And when we talk about new molecule ABC transporters, we find it out there are 59 ABC transporters that are present in ABC site, and they have a gene or gene segments duplication. They are widely expansion of A, C, and G subfamily, reveal their importance in the mosquito biology, and suppressor of immune ABC transporter is the survival strategy of the virus. There are different scopes. We can also work on the antigen specificity of innate immunity, suggesting to explore the list of transporters and other immune molecule genes in the regulation of virus development, and designing the strategies to manipulate this important molecule for blocking the pathogens development and transmission. So I would like to stop over here, and I would like to acknowledge the stat work is done by Ruti Kushar, Apple Coco by Vicky. Professor Sanjeev is with us, doing all sorts of experiments and helping the students, and whenever we stuck, this is my team, here's the ventilation is working on this work, and this is students Pushpata and Jyoti is working on the ventilation. Thank you.
So, no science is a complete science, that's why it's keep on going. But still, if you have any question on the halfway science, please raise your hands. Yes.
I have a couple of questions, so very interesting. First, have you tried silencing the transporter in selection plans?
No, it's just like first of all, like you have to target one organ. So we target the gut, salivary gland we haven't tested.
The other being, so initially, I think the bacterial infections you mentioned, you were using a mixture of live and dead bacteria.
Yes.
Is there any specific reason?
No, actually, it's not very specific. We just would like to say as a antigen elicitors, how they are working. So if it is a dead field, that means it's going to be molecules when we are using live, what there is that is.
Any?
So Lolita,
Yes.
good lecture, good piece of work. Just couple of observations or questions, whatever you can say. You have done work on ABC transporters,
Yes.
right? So is it possible at this juncture, can you correlate the dynamics of this ACP transporter genes with the propagation or development of this pathogen, for example, in malaria mosquito you were talking, or you can say their transmission? I mean, can it be correlated with the development of the pathogen and its transmission, transporter genes and the transmission?
Okay, sir. In that case, like we started with the first to align all these ABC transporters present inside the mosquito. And first we find it out like there are so many duplications over there, and almost they have like one say ABC A, there are also several ABC A transporters. Some of them we are targeting, like we just talk about G. We give the bacterial infection, and yes, there it's showing, the science is showing, yes, it is working. But further, we have to do more experiments
Okay.
to find it out how to control it. Antigen lowering bias.
Okay, just give you one or two ideas.
Yes,
In Singapore, there was a dengue problem.
yes.
There's one company, Oxitec, they released transgenic insects.
Mhm.
So I think if you can catch hold of those people, I mean, you can see these transporter gene mechanism in those mosquitoes,
Yes.
which have been released for that purpose. Basically, the concept is based on sterile insect technique. And for the last 3 decades, I have been working on that. So my interest is, if you can correlate that, then I would come to the next question, whether the radiated insects can have some sort of, mechanism working over there or not. And another point is that suppose you are able to find out this innate immunity, will it be possible that you can use some efficient method to, block the transmission?
Yes, sir. In case of like plasmodium, when we talk about plasmodium, we are looking for the molecules, and especially when we are doing salivary glands. So in the salivary glands, they are finding out the sporozoites during that time and how they can be able to block the transmission. There are several molecules which are working.
Thank you. I don't know whether you are remembering or not.
Yes, sir.
Someone got the professor's position. I was incidentally the expert in that committee. So I have got another connection with these people.
Thank you, sir. Please, ma'am, as the first part of your work, so any of this add inhibitors can also be able to molecule to inhibit the virus. What sir is asking to block it.
Sir, actually, when we look for the stats in the impact, we find out the NOS. And NOS is really this very well known, unlike the Professor Sanjeev is also working on product studies and NOS. And that is we are planning to really look for that.
Plant extract you are showing that has some ovicidal property also. It can be at the egg stage.
At this stage, we haven't checked this. Yes, we haven't checked it. But of course, it has a mosquito stage, but not egg stage. We have never checked it.
Check only the larvas.
Larvas.
And it can.
Because it has ovicidal property.
Yes.
The other one.
Now they have seen water extract, they may see different types of extracts,
No, whatever the extract we tried, it
Works.
but we only stick to the water one. Because preparation is easy, and we are avoiding the chemicals. So water one, the
No, water extract is fine.
But he is looking for
Instead of
egg.
larvicidal, whether it has ovicidal property, means you can hatching up, it can be avoided. No?
Yes,
You
you
want-
want try it.
We can try that.
That is more advantageous, more preferred.
I think there are a couple of things very interesting one can do. I can give one idea at this moment. I may not forget that. There are certain photosensitizers and they are very well documented, but very less work has been done in that case. So photosensitizers are very safe. At the same time, India has got no dearth of sun. The moment you use those chemicals and there is sunlight, they will immediately, they'll be toxic. Alpha T has been, it has been proven a very good insecticide against these mosquitoes as well as Lepidopteran insects. And you have been giving the data about Bombyx, and these are only two groups, Lepidopteran and different. Photosensitizers का थोड़ा सा देखिए, उसको इसके साथ जोड़ेंगे, it will be great.
Thank you. Thank you.
Thank you, Professor.
I request Professor, please felicitate Professor Lalita Gupta for delivering such an exciting lecture.
was absolutely yes.
Start with the next. Okay, we have another candidate from our department, very brilliant candidate. Dr. Soma M. Gorai. She is a strict professor in Hindu College, and area of interest is comparative immunology, microbiology, biochemistry. She has been a wonderful student, and she got this research associateship from DBT CSIR, and very sincere student. Right.
First of all, at the outset, I'm blessed to be here to be chaired by Professor R.K. Seth. He has been my teacher, and it feels great to come back to the department once again. And again, I would like to give my utmost gratitude to Professor Alok. When he just popped up this question to, have me to speak something, I said, this is not exactly my field, I work on reptilian immunology. But then, yes, the COVID times has taught us many things, isn't it? So those 2 years of the college where we had nothing to do, but we had certain, the worm inside our head, and we wanted to do something, not to sit idle and do something on nutraceuticals. I collaborated with a few of my colleagues from chemistry department, and they were synthesizing some novel compounds. And those are natural compounds which are usually found as herbs and spices, the main compounds. And those being chemistry people, they were actually looking into for the green synthesis. The way it's like green synthesis means like the way you synthesize some certain compounds with the minimum use of organic chemicals, so that can be easily metabolized in the environment. They are safe to consume, and they have drug-like capacity. So thanks to Professor, she's Dr. Neha Kapoor from my chemistry department, and we both worked on few things. And as I said, when the topic of this conference was told to us, it was nutraceuticals and chronic diseases. And COVID is one thing that we were actually going through this, and we just thought of working on certain nutraceuticals and how they would work in COVID. So we all know, yes, nutraceuticals, and it has been told for the past 1 day that we've been listening to so many people talking about cancers being taken care of and other diseases like leprosy. I just heard Professor G.P. Talwar in the morning. These are the chronic diseases that are being taken care of, cured by nutraceuticals. And yes, here we are talking about viruses, because even these nutraceuticals have certain role in virus curbing of the virus. And here we have certain basic spices that we usually use in daily lives, like cinnamon, turmeric, black pepper, basil, giloy, neem, garlic. It's everyday use, and they also have antiviral activity. So looking into that particular thing, it's purely in silico work. As I just said, it is a purely in silico work, where we looked into for few important components like the cucurbitacins, vixitin, berberine, mangofuridine, these are a few compounds which have been, there are many papers by the time, quite a few papers have already been published stating their importance in having certain antiviral activity towards COVID-19 or SARS-CoV, I would say. And we listed about 32 compounds from the literature, and these compounds had certain drug-like molecules, and they can actually have they can show the effects on 4 or 5 main targets that are there for the COVID infection that can be caused by COVID infections. One is the proteases, that is the Mpro, and these are the replications of the polymerases, the helicases, and yes, of course, the non-structural and the structural proteins that makes the entry into the cell, the virus which makes the entry into the cell, as well as As you all know, the cytokine storm that is released after the infection, and how to curb the cytokine storm. And these cytokine storms are basically taken care by the JAK-STAT pathway. So basically, all these molecules can be targeted at any of the levels. So this is a common diagram that also is made by one of our, this is an amazing thing. The kids these days are very, very fast. So this one of our biology student who prepared this graph, and she made this and it was published now. And this work we actually did with cucurbitacins. And I'm not presenting that work here. It's the work we did with cucurbitacins. And we could show that cucurbitacin H, G, and G, that is a glyphosate form of the cucurbitacins that is found in the Cucurbitaceae family, would actually affect biotine silica in all these 4 clusters that have been shown. The one, it could affect the entry point here, it can affect the proteases, it can affect the polymerases, as well as the helicases, as well as can affect the JAK-STAT pathway. So we went further and looked for all the 32 compounds through the literature, and these are the 32 compounds, and all of them we went. We had this Challenger software with us, and the Mastro the software where we did the docking for each one of them. And here I can see it is done for the polymerases, and the best docking score was seen by these 5 important components: actin, beta-lactam, butane, emodine, and curcumin. Apart from the other ones, they didn't have that better score. And we went on further. We did again the dockings with all the 32 compounds. With this is done by the human AC receptors. The receptors, which is we all know where the entry happens. The spike proteins basically bind to this, and the entry happens. Here also, again, we got the repeated answer of having the actin, beta-lactam, butane, emodine, and curcumin showing the best score. We went further, and then we did again with all these 32 compounds for the protease, the polymerases, as well as the JAK-STAT pathway. Here we could get butane as one component being a common among the others, whereas for the JAK-STAT, yes, of course, we have the emodine, the beta-lactam, curcumin, as well as butane showing. So here, in all the 4 cases, butane is one common component which has been repeatedly showing itself, and it has been able to show a good, beautiful D-score value with all of the PDB files that we could do, and did the scoring, the docking studies. Further, we took this molecule and did the further activity. We went on for checking the ADMET. Now, this is ADMET is done to check the drug-like capability of any molecule. So this is again software where we see the ADMET stands for absorption, distribution, metabolism, and the E stands for the toxicity and all. So these components are looked into there are many components, out of which it's a rule of Lipinski 5 rule which has been followed. What is that? Lipinski means where there are 5 important characters which every drug should, they should be able to follow that. If they don't follow those important rules, they do not fall under the drug-like capability. One is that it should have less molecular weight, less than 500. They should have hydrogen bonds, rotatable hydrogen bonds, acceptor hydrogen bonds, and there are certain, this is the lipophilicity and the distribution, this is the log S value. Log S value shows the flexibility of the molecule to dock. So basically, these are the important 5 Lipinski rule, which these are the basic steps that we go in before we actually take any molecule to in vitro and in vivo studies. So these are the basic steps, like looking in for the docking studies and then going in for the ADMET studies. So here also we could see that actin failed in 2 main important components. It failed in the molecular weight as well as it failed in the hydrogen bond. If you have too much of hydrogen bonds, that also makes it a non-good drug because that loses its flexibility. The flexibility has to be in a manner that they can actually undergo certain conformational changes, because whenever any receptor binds to a ligand, it needs to undergo certain conformational change, only then they can go on and activate the or do the signaling pathway. So the flexibility of the hydrogen bonds is also very, very important. Apart from the hydrogen bonds, there are other hydrophobic bonds, van der Waals forces, as well as the non-ionic forces, which are also taken into consideration. So this we did detailed molecular MD simulation activity, where this first we did with the HC, the human HC molecule. Here you could actually see this is made up of basically, there are two main important subdomains. This is subdomain A or 1, you could say, and subdomain B, which is separated by, you can see the yellow helix. This comprises about 17 different amino acids, and this is this we basically did with butane, but this applies to all the small molecules that can easily dock to this particular HC molecules, which shows that this area is where basically any molecule which has molecular weight, comparable molecular weight, can dock to this particular region. And this basically shows that light blue meshwork shows that this is where the binding of any ligand takes place to the HCE, that is the human ACE2 receptor. When I carried out, when I go to this region, this is the part which shows quite a good docking, and this one. So little over seconds, this was done by MD simulation. Now what is MD simulation? MD simulation is something which looks into protein-protein interactions, and where we're looking for a more closer look into how the interactions are happening and at which bonds it is forming. So when we go into deeper, this is the structure of butane. Basically, this structure is a simple structure, and it can be easily characterized, it can be easily taken, and butane can be easily synthesized in the lab. For that man, it is almost found in Butea. Butea is Palash flower. I think that is also known as forest of fire. During the monsoon seasons, Delhi was a full weight monsoon, that big Palash flowers, beautiful red color, yellow color, it's orange color flowers. And butane is also seen in some other, butane is also found in most of the yellow and orange color flowers. So even you can find it in gladiolas, and even in the sunflower, the other, I would say chrysanthemum, the orange color chrysanthemum also has that butane molecule. So here when we say the interaction, we could see that the lysine, the asparagine, the tyrosine, and proline and aspartic acid. So basically the hydrogen bonds are same with the asparagine, tyrosine, aspartic acid, and proline. And this non-ionic bond, which was seen with the, this is basically, I would say, this is the non-ionic bonds, or the butane, which is not a hydrogen bond basically. So this, there are many electrostatic bonds also, which is important during the binding of any ligands to the this thing. So these four important moieties are, we can see where any molecule, it is, butane is something that we are taking into consideration, but these are the basic moieties, the amino acid moieties which actually bind to the ACE2 receptors. Further, we did MD simulation. Now this simulation is done, this is done by the simple software, which is known as the, it's known as the web server, exactly what, it's known as the Haddock web server, or where the entire study is done in nanoseconds. So it starts from 0 nanoseconds to 500 is the maximum limit that is taken care of, where we actually see the stability of both the ligands. Now this designates butane, and this designates the HCE. Here till 200 nanoseconds, you can see the stable condition, and later on from 200 to 500, you can see certain peaks that is happening here. So here certain flexible or movement is seen here. Now this is basically considered by two important methods. One is known as root mean square deviation, and the other one is known as root mean square fluctuation. Deviation and fluctuations means how they are changing the conformation. Deviation, if it is less deviated, that shows that stability, and fluctuation is also very important. it's not always the stability that matters. Fluctuation is also important because that helps in changing the conformation of that area. This is very important because allosterical binding of any molecule is important because that changes it, the conformation, and helps like if you are using a small molecule and acting as and looking at as a drug-like property to bind to the ACE2 receptor so that it doesn't go and bind to the spike protein, it is important that they allosterically bind to the area, change the conformation, and not let the spike proteins to bind on it. So it's also important to have the fluctuation thing. So later on, when we did the fluctuation, we can see certain peaks happening. You can see certain peaks. And with further investigation later on, there are other software known as Haddock. This is HADOCK Haddock software, which takes care of how to looking for these fluctuations and how actually they are doing the changes in the conformation change. We did this thing, and we could see that at 0 nanosecond, the blue color, the cyan color, has the docking is like this, the binding is like this, and after 500 nanoseconds, it has come to this yellow color docking. And you can see the conformation also has changed here. And now there are only two important areas where it is binding properly, this and this. Rest other has undergone certain conformational changes. So this was further taken care of. We did a little more studies, and here we looked into how basically the original SARS-CoV-2 spike protein would have bound to the the human ACE2 receptors. We see that they bind basically at 4 main important domains, and these domains are known as hotspots. You can see that hotspot A, hotspot B, hotspot C, and hotspot D. So these are a normal spike protein of the coronavirus where it bound to the ACE receptors definitely at these 4 hotspots. Now, if we add butane, or here we are using butane, or the small molecule, the binding of the butane makes certain conformational changes wherein you can see only B and C still bound to them, where it has undergone not much of fluctuation as both the hotspot A and D. That means butane has bound more strongly to hotspot A and hotspot B. And that's what these fluctuations all show. These fluctuations, as I said, these all peaks, these are important because these fluctuations has led to the conformational changes at these 2 important hotspots and made the binding of the butane more strongly to the ACE receptors, not letting the spike to bind to it. So this is how it shows it. And further, we could gather, we went a little more ahead with the MD simulations, and we could see that these hotspots bound to be mainly at the tyrosine, at the then it is this is the lysine, this is the isoleucine, and then there's a threonine, and then there's this aspartic acid. You can see this blue color shows the hydrogen bonds, and these ionic bonds are almost nil. So you can actually imagine, as I was talking about, that K144, that ionic bond that was already there, in between, that has been totally taken care of. There is no ionic bond. It's basically all the hydrogen bonds and the hydrophobic bonds which has made the... now, when we talk about the bonding patterns, it's very important that the hydrogen bonds have to be moved. And whenever there's a hydrogen bonds or hydrophobic bonds in the form of van forces, they make a much stronger association. Ionic bonds makes it a less associated one. So it has been seen that we almost have no ionic bonds and good amount of hydrogen bonds. And these darker hues shows that multiple places that they have binded to. It's not, see, it's the binding is in nanoseconds. It's a process where it's the entire thing happens like it's a reel of thing that is happening. So when it starts from 0 nanoseconds and go to 500 nanoseconds, they are binding and releasing themselves. Again, they are binding and releasing themselves. So, but the way they bind to different moieties, that is important. So when we see this, the certain important amino acids that are taken care of and that they are binding to that particular thing. That is what makes it important, and that also says that how this introduction of small molecules which can bind more strongly to the HC receptors, not letting the spike proteins to bind to it. So basically, when we come to the conclusion, I will say HC receptors have two main important domains. One is the functional domain, and other one is the regulatory domain. SARS-CoV S1 proteins definitely bind to the functional domain, and this participates in the entry part. And if the small molecules goes and binds to this functional domain, it acts as a modulator. So these modulates the binding capacity of the spike to the ACE2 receptor. And we have seen that butane, the entire docking score of all the things is not more than 3.86 kilocalorie per mole, which is the perfect docking score. Anything beyond 5 is a good score, and it has almost 7.38. And it also follows all the 5 Lipinski rule of the ADMET. And we can also see that it makes good connection with hotspots A and hotspots C, as I have already shown it to you. That means it makes a good connection with good fluctuation points at certain important amino acids, letting not these spike proteins bind to it, which is basically binding to the hotspot B and C. And thus, I can say the butane makes a conformational dynamics change and helps in it can be used as a drug molecule. Now, what are these? These are nutraceuticals, and these are at a very early stage. We just talked about them. It can be taken up like now when we are back to our labs and all. We are now hoping to make these molecules and look it into how they actually going for the biological efficacies. Now, whether they have antiviral or antifungal activities or anything else, that has to be checked. But before you go for any of these biological activities, the first thing that with any molecules which has been synthesized or been characterized or by being taken out from any natural sources should be first let to see whether they are actually acting as a drug-like component or not. So this is my work. It is very small, a very crisp work during this COVID time, and this is the paper that we had just published in Journal of Molecular Modeling. I am thankful to my team here. Neha is the one who had given me all those components, the 32 components, and we had worked together. And the later part of the MD simulations, we couldn't find anyone in Delhi. In fact, we got help from somebody. It's Simon, who works in. He's in Pisa University in Italy, and he relentlessly helped us in doing the last part of the MD simulation, because we wanted to actually see how much docking study and how much the binding of and which are the major important amino acids are there in the MD simulation. Without the MD simulation, we couldn't have come to the conclusion. So I thank my team, and this is the paper that we had just published. And it's, of course, it is almost negligible to the work Dr. Bhatt and Dr. Lalita said. They have done a very wonderful work, and my work is just a little bit, which we could do it in the times of pandemic. And thanks for your kind listening. And we also did some work with the cucurbitaceous. I didn't show this here.
One should never be proud of itself, but to the work they have done. So always feel proud of your work.
Yeah, but yeah,
Whatever
definitely.
you have done, feel proud.
Yeah.
Any question, please?
Nice presentation, ma'am. Ma'am, we have done the docking with the ACE2 receptor, and we know that spike protein is continuously changing by the mutation, so that's why number of variants.
See, I tell you, coronavirus as such, see, all these studies, when we work on all this, the PDB files that we got, it's just not today's work. SARS-CoV was there from 2010 onwards, and people have been working on them. Many papers are there. So all the PDB files or CDX files that we have retrieved from the literature was basically based on the work that was already been done. So there are many important proteins, there are 3 important proteins, SME, out of which S1 proteins does undergo mutations, but the main important proteins that are involved in the docking are still very stable. Sorry, sir, मैं जरा उनसे बात कर रही थी. वो बच्चा पीछे. Okay, the other thing that the major mutations that have taken place, if you can read through the literature, it has taken in the non-structural proteins. Okay, so these are NSP proteins and NSP13 which have gone so through. NSP13 is one particular protein which has undergone much surgery. बच्चा को बता रही थी. So that is the mutations. So when we talk about the Delta and the Omicron variants, that is where it is. The spike is still the same. And there are, if you go through all the literature, all those 4 hotspots has not changed. Surprisingly, it hasn't changed. So the changes are there in the other non-structural. There are 17 to 19 non-structural proteins in this thing. So whenever you are looking for a drug molecule, you have to look for those proteins which has maintained the structures. So SARS-CoV has been always there. The work has been all the competition work or all the works that has even can Bhattacharya can say this conclusively. Most of the work we do is already based on the work that has been done on SARS-CoV. SARS-CoV-2 is the recent one, but the structure has not changed. Okay.
Was your question complete? That's what the question was asking, or something else?
I'm just curious about that part, that spike is continuously changing. So maybe that compound which is showing in the docking result it bind to the binding sites and inhibit the binding with the exclusion. But when the mutation is happening in this,
She's saying sir.
maybe that binding site due to structural changes get altered. So that's in that condition maybe that compound is not able to bind.
Yeah, that's what I said. No, the spike is not changing. That's what I said. All the 4 hotspots is not changing. That's still there. That's what I am.
Molecule is binding to human H2.
Yeah.
So that molecule will still bind there.
Yeah.
But your concern is also valid.
Yeah, definitely.
Because now changing the thing, mutations are happening.
Yeah, definitely.
So the drugs will definitely, sensitivity to the drugs will definitely change.
One suggestion I would like to give. It's good that you are interacting with your colleagues and it's a good work during that pandemic time. Do you have any plans to interact with people where wet?
Yes, sir. We are doing it now, sir.
Because that's why.
So yeah, we are now extracting the
modeling studies.
Bilkul, sir.
And we hypothesize. But many things, microenvironment keeps.
Bilkul. So in vitro in vivo studies are important, and now we are.
Start on the other side.
So we have done already. We started, sir.
Any other question? I have a question.
Yes, sir.
In our body, situation changes, like sometimes we have high fever, sometimes low body temperature and so on, pH changes, glucose changes. Do you think these conditions can also affect the
Yes, sir. As I said, so whenever there's a binding thing happening, no, it's mainly the hydrogen bonds is important, and there's a small ionic bonding, which are also known as non-atomic bindings or ionic bindings. Now, these ionic bondings do affect through the pH and the temperature. And here you can see the bonding is only at one position. But there can be certain other changes because it's not very flexible. These are proteins and they are very flexible. So they definitely undergo changes during high fever or the change in the pH. I do understand. No, sir. Abhi to, sir, we have just seen the docking study and now we have started extracting it. And we are doing it a green synthesized, like it's been synthesized in nanoparticles and the green formula. So we are now looking into it.
Like there is no question, I think we should clap. Sorry.
You check the levels too. That is the addition to the answers of the map. Is already checked with the levels that is more favored and then more favored as changing the pH change or the other factors.
Actually, sir, he is right. Admit, अगर आप करते हैं ना, so actually they are not 5. We go by 5 Lipinski scales. They are about, say, I think Lipinski scale में कितने हैं sir? 5 तो हम देखते हैं, but when we do there are more than 10 other characteristics. So उसमें से आप 5 choose करते हैं. तो जो best होते हैं sir. जिसमें और वो सारे चेंजेस जैसे लिपोफिलिसिटी ऑफ कोर्स तो चेंज होगी आपकी पीएच से, और आपकी वो हाइड्रोफोबिसिटी वो भी आपकी चेंज होगी, तो वो तो होगी। पर जहां पर हम देखते हैं कि ड्रग्स 5 जो दे आर एक्चुअली एग्रीइंग विद दोस थिंग, वी गो फॉर दैट। सो दिस इज druggability quotient. It's known as druggability quotient. Yeah.
Well, thank you very much.
Yeah, thank you, sir.
I request Professor Sanjay Kumar for delivering such an insightful lecture.
Sir, there is a banner.
All the best.
So now among us, we have a very young scientist, Dr. She is an assistant professor in the Department of Computational Biology at Independence Institute of Information Technology and Development. Her area of interest is comics, data analysis, personalized medicine, drug design and discovery.
Thank you.
She is a PhD from Department of Biochemical Engineering and Biotechnology from Indian Institute of Technology, Delhi. She is recipient of Sargogan Khurana Innovative Young Biotechnologist Award, postdoctoral fellowship from Tsukuba, Japan. And now she is the assistant professor at Universe Institute of Technology. Dr. Justin, you have.
So very good afternoon, everyone, and thank you for the kind words in the introduction, sir. So Dr. Soma has already made my job much easier. So whatever she explained in detail is the initial part of my slides. So you will see a lot of repetition from what she has already talked about. But yes, what I'll be talking about today mainly deals with the anti-COVID potential of ashwagandha and honeybee propolis. And along with the in silico analysis that I'll be showing you, we have moved a step ahead and did some in vitro validation. And we collaborated with RCB to do some other viral-based assays, right? Just to start with. So just very general introduction. As we all know, we do not really need an introduction to COVID-19 situation. We all have experienced it in some or the other way during the last 3 years. But this figure shows you the most recent statistics. So the problem is not yet solved. So you can see over here, this is the number of cases that were reported in the last 24 hours. So this data is for October 6th. And this is the number of infections that were observed in the last 24 hours. And this is the total number of cases that have been reported so far across the globe. And it has already infected more than, more than 215 countries, right? And this is a brief introduction to the structure and genome of SARS-CoV-2. So it is mainly 29.9 kb in length, the genome. It is a single-stranded positive sense RNA virus, right? So mainly the third part of the genome, it encodes for 2 polypeptides, which encode for a total of 16 non-structural proteins. And these are some of the important proteins which are involved in the replication and the progressive infectivity of the virus. And this 1/3 part of the genome encodes for the structural proteins, which help in the making of the virion particles. And this is how the virus actually look like. So Dr. Soma has already talked about these different components, right? She mainly focused on these spike proteins. So we have this capsule, membrane, envelope, all these form the structural component of the virus, right? This is also something that she has already shown. So this is a schematic representation of how the SARS-CoV-2 infects. So I would like to, reiterate on this part. So for the virus to enter the host cell, these two host receptors are important. She mentioned about this AC2 receptor, and there is one more receptor involved, that is TMPRSS2, right? So this angiotensin converting enzyme 2, this is the first enzyme with which the virus particle interacts by the S1 subunit, and then this TMPRSS2, it facilitates the proteolytic cleavage of this spike protein, which then further facilitates its fusion with the membrane, and it is like endocytosed into the cell. Then this genetic component of the virus particle, it is released inside the cell, and using the host machinery, it encodes for these polypeptides that I was talking about, encoded by the 2/3 part of the genome. And there are certain proteolytic enzymes which are also encoded during this process, and they cleave these polypeptides to make the functional proteins. And during this part, this enzyme is formed, RNA-dependent RNA polymerase, which further helps in the replication and the transcription of the virus. So this is the general process, and after this, the virus particles, they keep on growing and like infecting the further cells. So these are the symptoms which are associated with this COVID-19 infection, right? And these during that time of SARS-CoV-2 infection, so these were some of the drugs which were repurposed, and they were shown to be effective against these molecular targets in the entire replication cycle of the SARS-CoV-2. Right? So for the work that I'll be talking about today, I'll be mainly focusing on these two as molecular targets for ashwagandha and honeybee propolis-based bioactive compounds. And along with that, I'll also be talking about this 3CL-Pro, which is also known as the main protease of the SARS-CoV-2. Right? So as I said, we are targeting TMPRSS2, which is transmembrane protease serine 2, then AC2, this we have already talked about, and the main protease or the 3CL-Pro of the SARS-CoV-2. Right? So why ashwagandha and honeybee propolis? So this is the part of the work that I'm presenting today. This was done as like during my postdoctoral fellowship at AIST in Japan. So the lab already works on these natural compounds, and we are looking into the anti-cancer potential of these natural compounds. But these natural compounds are also known for their antiviral activity. So there are many reports in literature that talk about the bioactives from this ashwagandha, and they have been shown to be effective against H1N1 virus, influenza, this herpes simplex virus, and even HIV-1 virus. So this was one of the important plants that could be looked into for some repurposing work, right? So these are the therapeutic activities which are associated with the ashwagandha bioactives, and these are the two bioactive compounds which are most abundantly present in these plants, right? So Vidoflurin A and Vidinol. So not only these two main bioactive compounds, but there are many other different vidinolides which can be used from ashwagandha for various therapeutic activities. So for our study, we looked into these small molecules which are extracted from ashwagandha and are abundantly present in their extracts. Along with that, this is another small molecule that has been used. I'll tell you the use of this in the coming slides. So first of all, we wanted to see for the preventative measures of these ashwagandha compounds. So for preventative measures, we wanted to see if it can somehow help in reducing the interaction between the spike protein and the host receptors, TMPRSS2 and ACE2. Right? So to start with, we started with TMPRSS2. So the TMPRSS2 structure is not available in PDB. So the first thing was to model the protein so that we can look into some structural aspects of interaction of these small molecules with the protein. And the model structure is also available in the Swiss model repository. So it is modeled using the template serine protease hepsin of human. And the identity between the human and like the template and this TMPRSS molecule is 33.82%. And using the structural studies and some reported literature, it was found that this domain, which is actually involved in the priming process of the spike protein, it is highly conserved. And these are the three catalytic residues which perform this priming or cleavage process. Right? So this histidine 296, serine 441, and aspartate 345. Right? Now, when we are doing docking studies, now the theory behind all these experiments have already been explained by Dr. Soma, so I'll not get into the detail. I'll just touch upon those and I'll come to the results. So when we are doing docking experiments, the docking score that we get, they are not absolute scores. So they are always relative, like we can use them in relative to some known molecules. So this chemostat mesylate is one of the known molecules which can inhibit AMP-RSS2. We have experimental evidences for this, and a lot of experimental reports are also available. So this molecule, it is clinically approved and it is also extensively used for the treatment of oral cancer and some chronic pancreatic disorders. And recent reports, like the reports during this COVID time, they showed its importance for controlling this TMP-RSS2 as well. So the first, and its molecular mechanism of inhibition is that it interacts with these three catalytic residues, making them unavailable for interaction with the spike protein. Right? So the first thing was to dock this chemostat mesylate into the same pocket and see what is the interaction energy or binding affinity of this particular small molecule with our target of interest. So chemostat mesylate docking, and we got a docking score of minus 5.9 kcal per mole. So this acted as our reference or control molecule for the in silico analysis, right? Similar to the in vitro experiments, we do carry some control or reference molecules for the in silico analysis also. So this is one of the known inhibitors of TMP-RSS2. Similar to that, we used these different vidanolites, which I showed you in the previous slide, and we saw whether they can mimic the same kind of binding behavior against TMP-RSS2 or not. Right? So if you look at these figures, these yellow dotted lines, they show you the hydrogen bonds. And apart from these major hydrogen bonds, there were many other residues which were helping this ligand to stably interact with this pocket with the help of hydrophobic and wonderful interactions. They are not shown over here because they are lot in number and the image would not have been clear. So for vidofludimus A, the docking score was minus 5.6 kcal per mole. And you can see that this molecule, because I'm not showing you the entire protein as such, so this is the zoomed in view. So that these all molecules are interacting in the catalytic pocket that I showed you. So that is evident by this serine 441. So you will be able to see this in all the figures. So this molecule was interacting in the way similar to the chemostat mesylate that I showed you in the previous slide and is our control for these experiments, right? So vidofludimus A, then Vidanon over here, docking scores are listed below the images. Similarly, vidanolide A and vidanoside 5, right? Here you have vidanoside 4 and methoxyvidofludimus A, right? Then we have vidanolide B and 2-deoxy, sorry, I cannot read this name, 2-deoxyvidastra, sorry for the name. So all the docking scores you can see over here. So as Dr. Soma mentioned, she said that minus 5 is the relative scale upon which you can say the docking score is quite good. But yes, there are certain molecules which we have seen in our personal experience also, that even minus 2 docking score, but when it is in relative reference to some known molecules, they do give very good stable binding and they can also be translated in vitro experiments. So I would not say minus 5 is the threshold, but yes, you can always check in relation to the known molecular inhibitors. Right? So here, some of the molecules, they showed better binding affinity in comparison to this chemostat, but other molecules were also comparably similar, and the molecular mechanism of interaction was very similar to the known inhibitor, and mostly all of the molecules, they were stably interacting over there with the help of hydrogen bonds, right? But yes, as she mentioned this also, that docking, it generally represents only a static picture. So you just have a receptor molecule, you have the cavity in whatever form you are, like I'm using a model structure. So the cavity that I have just tried to fit that small ligand into the cavity in the best possible manner, right? But we know that in our body, everything is in dynamic motion. So that is what these MD simulations generally reflect. So what we generally do, we take these docked complexes, we put them in a box of solvent, which generally consists of water, and we try to see what happens to this over a given period of time, right? So as she mentioned, these molecules have been simulated for 100 nanosecond over here, because within these 100 nanosecond, we could see that the ligand is stably interacting with it, right? So she discussed about the importance of RMSD, RMSF, So here, what we are trying to look at, after the docking of the ligand with the protein receptor, whether there are some major structural distortions in the molecule or not, or is our new complexes showing a similar behavior to that of chemostat or not, right? So as you can see over here, the RMSD, initially deviates a lot from its initial dock pose, but during the time of simulation, it remains almost the same. Right? So it takes some time to adapt to its, most optimal conformation, but after that it stays in the same state. So this overall represents the stability of the protein ligand complex. But over here, this is only for the ligands. So it is possible that the ligand, like we generally see that sometimes if the binding is not that appropriate, the ligand leaves its original place, it gets and unspecifically, goes and binds to some outer surface of the protein and it stays there stably for the entire simulation. But here the, stable RMSD for the ligands alone, they were also telling us that the ligands were stably bound in the initial place where they were docked, right? And again, the RMSF, it did not show any deviation in comparison to the complex with the non-inhibitor, that is chemostat. And hence, the overall complexes were very similar to what we were looking for, right? So this is what we initially analyzed from the molecular dynamic simulations. And yes, the interaction profiles that she was showing. So over here, the green represents the hydrogen bonds, which define the specificity of small molecules towards the receptors. Then the hydrophobic interactions, yes, they are also a very important component when we are talking about interactions or stability of these small molecules in protein complexes. Some ionic interactions, but in most of the complexes, like in all these complexes, we will not be able to see these interactions, and some water-mediated interactions. As I said, simulations are carried out in water boxes, right? So this is the profile for chemostat mesylate, right? And this shows, like I said, the simulations were carried out for 100 nanoseconds, right? So if we consider the entire time frame as one fraction, so this shows you the fraction of time for which a particular interaction with a residue was important. So here the residues are not very clear. We need not bother about it for now. But yes, the interactions, these were the interactions and this is the fraction of time. So though the fraction of time maximum should be 1, but you can see up to 2 over here because one residue could be involved in more than one hydrogen bond formation. Right? So similar to this, if you look at the interaction profiles of the vidonolides of our interest, so the interaction was always better than that in the chemostat mesylate, or it was comparable to that. So yes, whatever we are, thinking of right till now, that these molecules can show similar behavior in comparison to chemostat is still, with us, right? So, and after this, now we were confident to take these to the in vitro analysis. So the first thing was to look at different cell lines. Now, these molecules, because as I said, we mainly work with antiviral potential, like looking into the antiviral potential of the nutraceuticals, right? So we looked into the expression of our molecular target of interest, that is TMPRSS2, into different cancer cell lines, and we chose the cell lines which express TMPRSS2 from moderate to high levels. And I'm showing you, these are the representative figures only, but we have done the study in multiple cell lines to be sure of what we are looking at, right? So these are the different vidinolides, like the numbers are representing vidinolides, what dosages are being used over here, and this figure shows you the effect of these vidinolides on the mRNA level of these different, like in TTN cell line, the mRNA level of TMPRSS2, what has happened to that upon treatment with these different vidinolides. So as you can see, most of the vidinolides could downregulate the mRNA levels of TMPRSS2, right? And this is in TTN cell, and the this panel of figure, it is showing you the same mRNA levels, but in the different cell lines, and using the vidinolides, which showed a better, or you can, which showed a significant downregulation of mRNA in the first set of screening, right? So TTN and HSC3. So the doses that we have used for the treatment, they are all non-toxic to the cells because we have been using these compounds from quite long now. So for all these cell lines, non-toxic doses have already been elucidated, and we are using the same. So these all, they do not kill the cell lines as such, right? And not only mRNA levels, we also looked into the protein expression of TMPRSS2. So I'm only showing the data for HSC3, right? So as you can see over here, even for the protein levels, there we could see a decrease in the expression level of TMPRSS2, right? And the same has been shown using like ICC also. Okay. Now, what I was talking about in these slides were like pure compounds which have been isolated from ashwagandha, right? But what if we use extracts as such? So, Dr. Renu and Dr. Sunil Kaul's lab, they are also working on optimization of extraction of vidanolides from these ashwagandha plants. So they have actually seen that using cyclodextrins, most of these vidanolides can be extracted in high concentration, right? So using water-based and like water-soluble and water-insoluble components, so they have created these 4 different extracts from the leaves and the stems. So the first one is the water-soluble like components of the like ashwagandha, and the lower one will be water-insoluble components of ashwagandha. So these 4 different extracts shown over here by 10, 11, 12, and 13. And this is the percentage amount that we have used from the extracts that were prepared in the lab. So as you can see over here, these extracts were also able to show similar kind of effect. mRNA levels were also found to be downregulated, and protein expression also went down. And the same thing would be seen in ICC also. So not only pure compounds, but ashwagandha extracts as such were also showing similar kind of results. Right? So this was about the first receptor, like TMPRSS2. This is the, like we also wanted to see what happens to the ACE2 receptor in this, the similar scenarios. So firstly, we looked into the residues of ACE2 that are involved in interaction with the RBD domain of spike protein for the docking analysis. So instead of blind docking, we would want to direct it to the structure or the part of the protein in which we are interested. So we created a grid over these residues and performed all the docking studies. I'm not showing the schematic representations over here, but yes, here we used this molecule as a control. So this is also a known inhibitor of like ACE2, and this has also been shown to inhibit H1N1 virus. And this is the docking score for this, and these I'm only showing you the residues which were forming hydrogen bonds. So these 2 residues were involved in hydrogen bond formation. And then if you look at the other vidanolides, a very comparable score, and even some had better docking score than the non-inhibitor. And these are the hydrogen bond interactions which were prevalent. And we did similar kind of MD simulations to check the stability of the molecules, and then whether we encounter some change in the interaction pattern or not. Everything was well conserved with respect to the docking studies. And we did similar kind of in vitro analysis. Right, so these are the different vidanolides that we looked into. So this panel shows you the effect on the mRNA levels of ACE2, and this figure shows you the effect on the protein, ACE2 protein, right? Similar ICC images, right? Then the effect of the extract, the 4 extracts on ACE2 mRNA and the protein levels, right? And this is the concentration of different vidanolides which were, found out using HPLC in the 4 extracts that I'm talking about. Leaf extracts, water soluble, insoluble, 10, 11, 12, 13 that were shown in those figure panels. And these are the extracts, the extracts prepared from the stem of ashwagandha. So these are the different vidanolides we could find out, and what was the concentration present in those extracts, right? Then, though these were the 2 receptors of the host cells, and we could do the in vitro analysis, but yes, we also looked in only the computational part of it, but we also looked if it can have some effect on the MPRO or the 3CL protease of the viral particle or not. So this is for finding out the catalytically important residues of MPRO. So this MPRO, or the main protease of SARS-CoV-2, it is highly conserved among various human and animal coronaviruses. So these are some of those. We did a multiple sequence alignment to look at the residues which are highly conserved and are also known in literature to play an important part in its catalytic activity. So the marked residues are the conserved one and were used for the docking purpose, right? And here the control molecule used was N3. So this structure is available in PDB as a co-complexed structure. So the same, we just studied the interaction of this N3 with the protein shown over here. So these were all the residues participating in the stable interaction of the small molecule with the protein, and was used as reference for comparing our compounds, right? So some of the vidanolides that I'm showing over here, their docking score and the hydrogen bonds that stayed after the MD simulations, right? So as you can see over here, this vidanoside 5, it showed much better binding with the Impro in comparison to other. But unfortunately, because of the being a viral protein, we could not perform the in vitro analysis using this. But yes, then we collaborated with RCB, Regional Center for Biotechnology in Faridabad, to do some viral-based assays. So here we provided them with these pure compounds, and they have taken this remdesivir as one of the inhibitors for the SARS-CoV-2, and they did some in vitro analysis. They co-cultured E6 cells with these different vidanolides and remdesivir as a control, and then they infected it with the SARS-CoV-2 particles, 0.01 multiplicity of infection, and then post 24 hours and 48 hours, they collected the supernatant, and they looked into the presence of E and N genes, and based on that, they calculated these percentage inhibition. As you can see over here, remdesivir, it showed the highest percentage inhibition of virus, that it could not get into the cells, right? Somewhere near to 99%. But if you look into the vidanolides, some of the vidanolides actually showed very good percentage inhibition. For example, over here, vidanolide B, right, almost 88% viral inhibition. And then similar to that, vidanoside 5, which also showed very good docking scores for all the 3 different components. Even in the in vitro cell line based assays, this was one of the most potent or potential candidate molecules. And though the range for the inhibition, it is varying for different components, like it is starting from, I would say, go for this 48 hours, like if I will say it's from 25 to even 84%, that is the perfect range. But when you are using extracts as such, you can see that the effect of these extracts could be very, promising. I would not say maybe curing the viruses, but at least for the prevention, they could be very good extracts, right? Yes. So similar to ashwagandha, we also looked into honeybee propolis. So Kate is one of the bioactive compounds from there. This is the molecular structure, and similar kind of studies, in silico studies, then the non-toxic doses determination, then looking into the mRNA levels, then the protein levels of the same. So yes, so all these studies also suggest that CAPE also has similar kind of potential and could be very much useful for the management of SARS-CoV-2. And all this work has, resulted in these publications. We started with in silico work, and we went on as and when we found more resources and opportunities to perform these in vitro studies, collaborated with RCB. Now recently, like Dr. Sunil and Dr. Renu Wadhwa, they have also signed an MOU with all India Institute of Ayurveda to perform more structured studies and look into the promising potentials and how it can be actually translated, like finding the better ways of using it and being more confident about what we have found in the laboratory. So at last, I would like to acknowledge all these people. I have a lot of people to acknowledge. So I'm as he said, I did my PhD from Dr. Sundar's lab in IIT Delhi, and it was that time I got introduced to the this group. People are present over here, Dr. Renu Wadhwa and Dr. Sunil Kaur. So this is my group at AIST. And now I have joined in IIIT Delhi. I would also like to thank them because they believed in me in my work, and they have given me an opportunity to now start independently. And yes, the group of Dr. Sudhanshu Vrati in RCB for performing all the viral-based essays. So that's all. Thank you.
Yes.
Yes.
And you mentioned like with all these compounds, they bind to ACE2 receptor and other receptor. But even mRNA expression is going down when you treat it with
Yes, sir.
What is the molecular mechanism?
So molecular mechanism is something that we do not know for the mRNA levels, but yes, we do know that it goes down.
Maybe you check the capacity activity, maybe ACE2 may be activated by electric capacity.
So, we do have a relation of, we do know that they do have an impact on all these molecules because, as I said, we are also looking into the anti-cancer potential. So there we have looked at all these markers, but correlating it with the COVID-19 for now, I cannot directly, say, because in context of COVID-19, we have not looked into it.
Last year we showed like that activate and inhibits CT2. And one of my students, she presented data about another phosphate.
Yes, sir. So the other reports from the group with the same molecules will have these markers in them, definitely. Yes, sir.
I think
He has a question.
As you said, showed in RCB some antiviral activity. So have you not checked it with Camostate decided there?
No, they have used remdesivir as a control over there.
No, Camostate decided, of course, not established as a positive control, but because it is one of the viral, because in my lab I'm not getting any activity with Camostate decided. That is one thing, and then second thing is Camostate decided already clinical trial was done and it failed. It doesn't work. So do you think that overall ashwagandha may be working as a good treatment for COVID, but not a individual
Yes,
molecules?
yes. So as I said, like the individual molecules, they have a wide range of activity from 25% inhibition to 84. So I would not say like pure individual compounds should be used, but extract as such. And we already are working on the optimization of these extracts so that we can have the good yield of these molecules in those. So based on that, we can look into it. So as market powder that is available, that is prepared using roots, but our analysis have shown that leaves have the maximum amount of ethanolites in those. So probably leaves are a better way of consuming ashwagandha.
We heard a very good lecture from one of the youngest speakers in this conference. Young scientist. Very good. Jaspreet, I just wanted to know one thing.
Yes, sir.
You have any plans to go for in vivo bioefficacy?
In vivo experiments are also done in collaboration with some other universities in the group itself. But yes, we do not have that facility as such at my place for now. Now I have moved from AIST a little. But yes, we do have plans to do that. Yes. Animal
And
models.
one thing you said about the propolis.
Yes.
I was expecting some, data from that also.
Propolis, actually, I just skipped because the
I
study.
understand.
Yes,
There was a
the data is
limitation of time.
yes, but it shows inhibition over here, as you can see over
Being
here.
an entomologist,
Yeah.
I was very much interested to know about that.
So you can see over here, this is the iron level and this is the protein level. Both show inhibition. Yeah. Yes, yes. So that's why I just skipped it. I could hear the
You
buzzer.
mentioned about ACE,
Yes, sir.
angiotensin
Converting enzyme.
Can you imagine whether this ACE is present in arthropods and insects also?
It has some other roles also. If I can relate, it has some cardiovascular related roles as well.
So
So
by information they are found
so like could be present. Yes.
insects.
So maybe we can look in some other aspects of those as well. Yes. Thank you.
Thank you very much. It's brief.
Now I for delivering such a wonderful lecture. अच्छा
He's the last speaker, I suppose. Something do. I don't know whether I'm speaking correctly or not. He's associate professor and current affiliation is Center of Excellence in Molecular Biology and Regenerative Medicine, GSS Medical College, Karnataka. His area of interest is molecular basis of development and pathogenesis of obesity, autoimmune disease, and beta cell carcinoma. And he has got a vast postdoctoral experience in various places in the US, Virginia Commonwealth University, etc. And we are very eager to hear you, sir. I will request you to please go ahead with your lecture. Sorry.
Good evening, all. First of all, I thank organizer, including Department of Zoology, University of Delhi, and also the Society of Nutraceuticals and Chronic Diseases. And today I'm going to talk about the role of quercetin and how quercetin mimics vitamin D and eliminates breast cancer, into the hepatic inflammation and fibrosis. And I used to work with an animal model called early ascites carcinoma, and this is a mouse breast cancer cell line. It was developed by Dr. Paul Ehrlich. We all know that Dr. Paul Ehrlich is a Nobel laureate. He developed this cell line in 1907. And I used to work with this cell line during my PhD days, and I used to work with ashwagandha and Vitaferine during my PhD days. And I also published a paper. We treated Vitaferine to the CAC cells, and we found that ashwagandha, the Vitaferine inhibits sp1 transcription factor and inhibits the proliferation of this CAC cell line. And recently we published one paper where we showed that these early ascites carcinoma cells, they cause liver inflammation and fibrosis, and we published recently in Advanced Cancer Biology and Metastasis. And in continuation of this work, and this is regarding our research, and it is published in leading newspapers, including Hindu, Indian Express, and Star of Mysore, and all those papers. And what is the current objective of the study? Like our objective of the present study is to study the effect of like how breast cancer causes liver inflammation and fibrosis. And we all know that breast cancer is the most common cancer worldwide, and from morning we are hearing a lot about breast cancer. And breast cancer is a lifestyle and diet-associated disease, and also like except in few cases, it is a completely lifestyle-associated disease. And from our own study, breast cancer causes liver inflammation and fibrosis. Therefore, exploring the natural compounds and nutraceuticals with anti-breast cancer effects and hepatoprotective effect is our prime objective. And we know that people know the this quercetin. Quercetin is a plant pigment and flavonoid, and it is also acts as an antioxidant molecule. It is found in many fruits and vegetables, including apples, onion, mangoes, cherry, raspberry, and cranberries, and even in jackfruit. And it has very beneficial effects, and it is a potent antioxidant. However, the effect of quercetin on breast cancer induced liver inflammation and fibrosis is not known. Here we try to explore the role of quercetin on breast cancer induced inflammation, liver inflammation, and fibrosis. And we also elucidated the molecular mechanism behind this effect. And we used this EAC cells, and the EAC cells, they produce mouse mammary carcinoma, and they secrete a lot of ascites. These ascites contain a lot of angiogenic molecules and also a lot of inflammatory cytokines. And when we inject these EAC cells to mice, and they form a big tumor in the abdominal region, and mice will die in like 16 to 17 days or 18 days, they will die due to EAC cells, the overgrowth. And during this stage, we also observed enlargement of liver size and also inflammation of liver size. And to check the activity of quercetin, we treated quercetin to this EAC mice. And we observed after the treatment of quercetin, we checked the body weight, liver weight, and also tumor volume. And also we measured the cell viability and other biochemical parameters. And we also did immunohistochemistry to check what is the effect of quercetin on breast cancer induced liver inflammation and fibrosis. And we also used cell proliferation assay and CAM assay. Many of you may know about CAM assay, the Chick-Corean membrane assay. And after the when we grow EAC cells in mice, like you can see that middle picture where mice looks like somewhat obese, but this is not obese. This is due to cancer burden. And after the treatment of quercetin, this mice looks like this. This shows quercetin inhibits the burden of this breast cancer, this mouse breast cancer cell growth, and details the survival of these mice. And we measured the body weight, and we found that like when we treated with quercetin, there is a reduced body weight, and also we measured the tumor volume, and also the volume also gone down. And if you see this liver picture, you can see a liver enlargement. Like these ESC cells induces liver enlargement. After the treatment of quercetin, we saw somewhat reduced liver weight. This shows like this quercetin inhibits the inflammation of this liver enlargement and inflammation. And also we measured the liver weight. You can see gradual decrease in liver weight after treatment of quercetin. And we also measured cell viability. After the treatment of quercetin, we also saw less number of cells. And we also measured some of the major liver enzymes. These enzymes usually we use for liver function tests, AST, ALT, and ALP. After the treatment of quercetin, all these enzymes secretion goes down. And also you can see the inhibition of like when we induce ESC cells to mice peritoneum looks like this, and this shows lot of angiogenic blood vessels. After the treatment of quercetin, we saw reduced blood vessels. This shows like anti-angiogenic property of this quercetin. We also checked we did a HRD scanning. We checked like less microvessel density after the when we took this peritoneum of these mice. And we also measured the effect of this quercetin on early chassis-induced liver inflammation. There is a decreased inflammation and also decreased fibrosis. We also measured like inflammatory markers like TNF alpha, IL-1 beta, and also IL-6 after the treatment of quercetin. Even these markers also gone down. And also we measured TGF-beta pathway genes, including TGF-beta secretion after the treatment of quercetin. And we found that TGF-beta level was also gone down. And along with TGF-beta, we saw collagen 1 and collagen 3 alpha SMA level also gone down in these mice. And interestingly, recently I saw these two papers where we found that like tumor autonomous effects of vitamin D deficiency promote breast cancer metastasis, and also the role of vitamin D in reducing cancer risk and progression. Like vitamin D plays very important role. Usually, night shift workers, women, especially women, they get more breast cancer. And also due to change in lifestyle, a lot of people nowadays, a lot of young women are getting breast cancer. And vitamin D plays very important role in prevention of breast cancer according to these two papers. And also like many other papers also showed that vitamin D prevents breast cancer. We got some interest on vitamin D, and we thought of looking for vitamin D receptor and what is the effect of quercetin on vitamin D receptor based on this background. And we have some collaborators in our JSACHR. They did tracking studies for us, and they checked the quercetin binding to vitamin D receptor, and they told that like quercetin very strongly binds to vitamin D receptor. Therefore, we thought that like quercetin may activate this vitamin D receptor and may target vitamin D genes like vitamin D. And we treated quercetin and vitamin D to two of the known breast cancer cell lines. Both similarly inhibits the proliferation of these two breast cancer like MBA, MB231 and BT474. And simultaneously we treated together like vitamin D and quercetin, and they act similarly. Like based on this data, we found that like quercetin maybe strongly activates vitamin D receptor. And we treated quercetin to chick chorioallantoic membrane, and we found that quercetin inhibits angiogenesis in chick cancer. And also we found that vitamin D also like similarly inhibits as quercetin. They both acts in a similar fashion. And we also did CD31 staining to peritoneum of these ESC mice after treatment of quercetin. we found that CD31 expression gone down after the treatment of quercetin. This shows that quercetin strongly activates vitamin D. And based on this preliminary finding, we propose this hypothesis where quercetin binds to VDR and it activates vitamin D response element and induces vitamin D related gene and inhibits the proliferation, inflammation, and fibrosis, and also angiogenesis. And one of my students, like Ms. Nirmala, she is working on the role of quercetin on obesity, non-alcoholic fatty liver disease, and cardiac fibrosis. Tomorrow she is going to present that data in our oral presentation. We have, obesity is directly associated with breast cancer. We also have the data like where the quercetin inhibits obesity and also cardiac fibrosis and also non-alcoholic fatty liver diseases. And she is going to present that data tomorrow. I thank my collaborator, Dr. Shashank Prasad, and also two of my students, they are here, and Ms. Nirmala and Ms. Varsha. And Ms. Varsha, she presented our oral presentation with aspirin and non-alcoholic fatty liver disease. And Nirmala, she will present tomorrow about quercetin and metabolic syndrome. Thank you all once again. Thank you.
Question from those?
Sir, does this molecule affect the hormonal level of ghrelin or leptin? Does this molecule affect the hormonal, just modulate the hormonal level of ghrelin or leptin? Because if ghrelin is more, then the obesity will increase.
We didn't check ghrelin or leptin, but adiponectin. When we treat quercetin, adiponectin level goes high. We know that adiponectin acts as an anti-obesity and anti-non-alcoholic fatty liver disease molecule. It also inhibits cardiac inflammation. She has data about adiponectin and quercetin. We have more data on adiponectin and quercetin, and also beta-ferrin, structurally it resembles steroidal lactone. Steroid hormones.
Yes.
Usually steroid hormones binds to various nuclear receptor.
Yes.
Based on that idea, we also did a docking study with LXR alpha and one more FXR and all those things. We found that beta-ferrin binds to these receptor and activates these receptor because it acts as like a steroidal hormone type.
Did you narrow down any particular targets for beta-ferrin binding to the receptor?
Yes, LXR alpha. We published that paper in Frontiers in Oncology last year, but still we have that data related to non-alcoholic fatty liver disease.
Very nice.
Okay.
We
We inject into peritoneum cavity, and also they also form solid tumor, but very commonly we use it as a.
If you could have injected in mammary to
That is what it is almost near to mammary region. It will form like a big tumor in abdomen region. This is liquid tumor, and we can also inject as a solid tumor along with the matricial. And many people they are doing even as a solid tumor model. Do you have any questions? Okay, thank you once again.
Now I request Professor Kumar and Professor Seth to felicitate Dr. Santakaradur for delivering such an enlightening lecture. Now I request Dr. Sonia Kaur to please felicitate Professor Sanjeev Kumar for chairing the session. Facilitate for holding the chair for this session.
Professor, entomologist. And Omir is also works on bioluminescence, so insects, indoors.
Fireflies.
Yes. That's right. That's right.
Now we'll disperse for a short tea break, and I think this session is over now. Thank you. Thank you.